A good way to think of the processes of mitosis and meiosis is if you think of them as being photocopied, except for during this process, they destroyed the original photo.
By the end of meiosis, you should have 4 haploid cells. So, during meiosis 1, you have the original photo, which comes out as 2 photos. You then have the 2 photos put in, come out as 4 photos, except this time, they're cut in half. So there's 4 haploid cells.
In this phase, he's living a pretty regular cell life. Eventually, the DNA curls tightly and you can see the chromosomes. These homologous chromosomes become close and eventually exchange segments by crossing over. During prophase 1, chromosomes combine to create homologous pairs. These homologous pairs then cross over each other to create genetic variants. During prophase 1, the nuclear envelope also disappears and spindle fibers can start to form. During metaphase 1, the homologous pairs are now lined up in the middle, and the nuclear envelope has fully disappeared. Anaphase 1, with spindle fibers shortened and dragging the chromosomes apart towards each pole. Both homologous pairs go with them. This means that the homologous pairs will complete a diploid set of chromosomes that has 1 chromosome from each homologous pair. Next is telophase 1, in which both new cells have 46 chromosomes. Then the nuclear envelope reforms around each set of chromosomes and spindle fibers disappear.
Mitosis has 4 phases and ends with cytokinesis, which is the final splitting of the cell membrane to form 2 cells. Before we can get into the stages of mitosis, however, I should talk about the phases of interphase. The phases are G1 phase, S phase, and G2 phase. G1 phase, which is right after mitosis, the cell rebuilds all its organelles and performs vital functions like building proteins. In S phase, the cell replicates its entire DNA via a process called DNA replication. In G2 phase, the cell builds centromeres and spindle fibers in preparation for mitosis. It's prophase. In this stage, the cell bunches all of its chromatin into chromosomes, and the nuclear membrane of the cell begins to disappear. Also during prophase, the spindle fibers begin to come out of the centrosomes. The second stage is metaphase. In this phase, the nuclear membrane has been totally dissolved, and the 2 chromosomes pair at the centromere to form 2 sister chromatids. These sister chromatids line up at the center of the cell in preparation for anaphase. In anaphase, the spindle fibers connect to the sister chromatid and pull them to opposite ends of the cell. In phase, the cell begins to split and the nuclear membrane begins to reform as the sister chromatids separate back into chromatin. It can be viewed as the opposite of prophase. The process of telophase and cytokinesis is helped out by vesicles filled with cellulose that build a new cell wall around the new cells to create 2 separate cells. This is needed because of the strength of a plant's cell wall. Cytokinesis is the process of splitting the cell membrane or adding another cell wall to form 2 new cells. Mitosis, the cell then goes back into interphase and the process repeats itself.